I can tell by the lull it's about time to start. So thank you all. It's just wonderful to see all of you here to the Wisconsin Alzheimer's Disease Research Center's 18th Annual Fall Community Lecture. How many of you have been to the fall lecture in the past? Oh, wow, that's terrific. What about first-timers? Group? I think it's 50-50. That's wonderful. I can tell all of you, whether you've been here before, you're in for a very special evening. A real treat tonight. The theme of this evening is New Frontiers in Alzheimer's Disease Research. And tonight we're going to hear about the link between diabetes and dementia, the connection between gut health and brain health, and that is a fascinating topic, and the evolving definition of Alzheimer's disease. For those of you who don't know me, I've been here before for several years in this position, but my name is Carol Kobe. And when I'm not here in front of you emceeing this particular fall lecture, I am producing and hosting the weekly radio program All About Living. And I like the number 18 because I also will be celebrating my 18th anniversary in producing and hosting the show this coming spring. So it's a very special evening for me as well. And like so many of you here, I have had my own personal journey with Alzheimer's disease. In the mid-90s, my husband at the time was diagnosed with frontal lobe dementia. And so I have been, like so many of you, in that position of spouse turned caregiver and watching on a daily and a monthly and a yearly basis the continuing new normal and the deterioration of someone suffering from one of the dementias. And it's heartbreaking, it's stressful, painful, angering, frustrating, all of those emotions, and the whole breadth of them that you go through that whole experience. Because as caregivers know, and the medical profession knows as well, the stress on caregivers is an extreme health risk for the caregiver, and not only for the person who is diagnosed with the disease. And having this personal journey with Alzheimer's and dementia really not necessarily changed my life, but it expanded my world and it gave me a better understanding. Soon after he passed away in 2001, I was invited to join the board of the Alzheimer's and Dementia Alliance of Wisconsin, and I eagerly agreed to do that, to say yes, because they had been so helpful to me along that journey. And I have been on the board and in other positions with the Alliance over the years and still remain so. But in addition, I was introduced to the wonderful work here at the University of Wisconsin, their Alzheimer's Disease Research Center, and the Wisconsin Alzheimer's Institute. And a few years ago, because of the growing work of Alzheimer's disease research here in our own backyard at our own University of Wisconsin campus, the University of Wisconsin School of Medicine and Public Health named the Initiative to End Alzheimer's Disease as one of the two major initiatives at the university. And this is because not only of the wonderful work they have done, but the global platform they have achieved as one of the leading global institutions researching dementias, looking for a way to prevent it, to hopefully effectively treat it, and most of all, to cure it. So when they started the Initiative to End Alzheimer's Disease a few years ago, they invited a group of community leaders and community people involved with the disease to join a board of advisors or visitors, and I was invited to be a part of that group as well. And so I am proud, privileged, and really humbled to feel a real part of what is going on at the university. I don't have the knowledge, the expertise, or the educational background to do the wonderful research and clinical work that they're doing, but I can speak to it from the community, from a person who has been personally affected. And it really is everybody's disease. It's a community disease, it's a family disease, and it's something that we all need to get our hands around and do something about, because so far the numbers are escalating in rapidly epidemic forms. So that's a little bit about my background of why I am here this evening. But on to our program. Before we begin, I'd like to personally thank all of the volunteers who have helped make this evening possible. And if some of the volunteers, you're scattered around the room, just wave your hand, and we'll give you a quick round of applause. As all of you know, it is so critically important to have volunteers to make something like this come together. You never know why it comes together so well, but it's always the people behind the scenes. And I hope that you had time to wander through our resource fair. We'd like to thank our exhibitors for participating this evening, and we thank them for contributing generously to the health of our community through the important work that they do. They are, in my idea, they're kind of like the Wisconsin idea, that it's the extension of what is going on here into the community, to serve the community with programs and resources. So we particularly appreciate them. Then, I'm sure you all received one of these red bags. Whoops, there's a lot in it. I'm not going to go through everything that's in it, but I would like to point out a couple of items to you. One of them is the yellow sheet, bright yellow evaluation form. This is really important that you fill this out. We will collect this evaluation form at the end of the program, and we hope that you will put in your remarks and your response to what you hear this evening, because it helps us ensure that we are bringing informative and relative programs to the community. So we greatly appreciate your feedback. Then if you're interested in participating in research, and that's another thing that I have joined with the university on. I am a research participant, and it's a wonderful experience. You feel like it's one way that you can give back. And there are many, many research programs at the University of Wisconsin in their Alzheimer's research program. So you have many choices to pick from, and they're looking for all genders, all ages, for many, many programs. And there's a brochure in your red bag. And if you're interested in just learning more about what it is to be a research participant, fill this out and either give it to one of the representatives when you turn in your evaluation form at the end of the evening, or send it into the university, and a research participant will contact you. And I assure you it's something that you'll feel really good about. And then finally, we will have questions at the end of the program, and you have these white note cards in your bag as well, and there's a pen, and so you can fill out questions that you have, and our staff will collect those at the end of the program after the presentations for the Q&A period, and then our panel of experts, our researchers, our doctors will be here to answer your questions, and they are very generous with their time, and they'll be happy to answer as many questions as possible. So remember to fill out your questions here. And then finally, just a housekeeping matter. You're hear this at all programs now. If you have a cell phone with you, if you could please turn it off right now. So that would be a good thing to do before we begin. And before I introduce our first speaker, there is a special bit of history that I would like to share with you. And the University of Wisconsin-Madison actually occupies Ho-Chunk land, a place their nation has called Dajope since time immemorial. In an 1832 treaty, the Ho-Chunk were forced to cede this territory. Decades of ethnic cleansing followed when both the federal and state government repeatedly, but unsuccessfully, sought to forcibly remove the Ho-Chunk from Wisconsin. This history of colonization informs our shared future of collaboration and innovation. Today, UW-Madison respects the inherent sovereignty of the Ho-Chunk Nation, along with the 11 other Native nations of Wisconsin. And I thought that's interesting for everybody to know. Please, it's worthy of an applause. Some things are especially poignant, and that one is to me. Our first speaker this evening is Dr. Sanjay Asthana. Dr. Asthana is the director of the Wisconsin Alzheimer's Disease Research Center. He is chief of the Division of Geriatrics and Gerontology at the University of Wisconsin School of Medicine and Public Health, was appointed the first associate dean for gerontology in 2015. He also holds an appointment at William S. Middleton Memorial Veterans Hospital, where he serves as the director of geriatric research. His father, an economist and diplomat for the United Nations, suffered from Alzheimer's disease. And that spurred Dr. Asthana's study of the disease. And we're so pleased because he is a wonderful and amazing leader and gentleman. So please join me in welcoming Dr. Sanjay Asthana.
You can hear me now? Hi, good evening, everyone. Thanks for being here. I want to welcome you all for coming here. And we are very excited to share with you some of the advances that have taken place in Alzheimer's and some very important areas of research that are being done right here at UW-Madison. So we have a full agenda for you. And usually they give me 5 minutes because they know this guy will go on for 2 hours. But I will keep up my promise. So what I thought that before I talk about anything in detail, let me touch about a breakthrough news that many of you might have heard yesterday. Raise your hands. How many of you either heard yesterday or today a new medication that has come out for Alzheimer's disease? And okay, thank you. So I thought maybe the very first thing to do is to just address this new finding that has come out. So there is a company called Biogen, and Biogen is one of the pharmaceutical companies that for many years has been doing research in Alzheimer's disease, and they wanted to find some effective treatments for the disease. So over the past decade or 15 years, multiple trials have been done with all kinds of medications, and unfortunately, all of those trials, at least till now, had failed in people with Alzheimer's disease. But Biogen sponsored a medication called aducanumab. I got it right. I've been practicing since yesterday, actually. Aducanumab is a medication that is given through intravenous infusion into your arm. It's given once a month, and it is an antibody that goes to the brain, and then it eats away that amyloid protein that we know gets deposited in people with Alzheimer's disease. So the theory was you give this medication as an infusion, it goes to the brain, and it clears the brain of this amyloid protein. And then the hope was that as we do that, perhaps over time the person's symptoms of Alzheimer's will improve. So they did this study for about a few years worldwide, and they did an initial analysis end of last year, and then they announced around springtime this year that the results of the initial analysis was negative with this medication, that it did not work. So they stopped all the trials worldwide with this aducanumab. However, what happened a few months later, that they got some more data of people who were still in the trial, and when they reanalyzed the data, they found, wow, actually this medication works. So they found that people who were in the trial, about 2,000 of them actually participated in the trial, and they found that there were improvements in their memory. There were some improvements in their day-to-day function, and also the amount of amyloid in their brain had really come down with this medication. So this is a very exciting finding in many ways. So they're going to approach FDA and try and get approval for this medication so that they can use it to treat patients who have Alzheimer's disease. Now, it remains to be seen if the FDA actually approved this medication. And it is a little strange that initially the analysis was negative, and then as they got data from a few more hundred people, they found this exciting result. So I think this is, for the first time in 16 years, that since approval of Aricept, raise your hands, many of you have heard of Aricept, the medication that is approved by the FDA, that we use for many years for treatment of Alzheimer's disease. For the past 16 years, no new medication had been approved. Now, there's a chance that aducanumab may get approved by FDA, and so we're going to keep our fingers crossed. But I think this is another testament and a proof that as we do research in this in this disease, we are one day going to indeed find a very effective treatment, new ways to prevent the disease, and one day a cure. As you know, there are no shortcuts to research. All the diseases we have controlled so far, diabetes, heart disease, cancer, is the result of decades of research on people with that disease. And now we are seeing some exciting results for Alzheimer's disease. So again, thanks for supporting. I know many of you are a part of the studies that are going on here, and he is a living testament that as we continue research, we will one day find treatment and cure for the disease. So very exciting result. We'll keep our fingers crossed and hope that the FDA is going to approve this medication. So with that, I want to introduce you the Wisconsin Alzheimer's Disease Research Center that Carol mentioned. As you know, about 10 years ago, the National Institutes of Health, which is the federal agency that funds medical research throughout the United States and actually outside the United States, in Europe and many other parts of the world, they gave us a grant to designate us as one of the centers of excellence in Alzheimer's research. In the United States, there are only 32 medical schools out of 148 medical schools that have got this designation of excellence in Alzheimer's research. And we are very fortunate to have that designation for the past 10 years. And earlier this year, in April of this year, they gave us money for another 5 years to continue with that designation and continue with research right here at UW-Madison. So that is very exciting. So what the ADRC does, it actually supports a number of studies. In fact, there are dozens of studies that are ongoing here, over 50 or so at any one time. And we're trying to find new treatments for the disease. We're trying to find what causes the disease. And we're doing all kinds of research, not only in people with Alzheimer's disease, but those who are at risk for the disease, and also people of different race and ethnicity, how Alzheimer's might differ between races and ethnicities. And in fact, today the keynote speaker today will highlight some of very interesting findings that have come out in Native Indian population. And our center is very focused in that area as well. Now, the NIH has received increasing support from the US Congress over the last 4 or 5 years, because it is clear to us that this country is facing an epidemic and a real crisis of the number of people who are being diagnosed with this disease. So a lot of money is coming from the Congress to NIH, and we are lucky to get a good lot of that money right here through competition that University of Wisconsin has received federal grants from NIH, and also there are a number of foundations. Alzheimer's Station is one of them, and many other foundations that support research nationwide and internationally, and donations from people like you who really, really support research for Alzheimer's disease. So the ADRC here is very prominent, is one of the very highly ranked centers in the country, and is renowned internationally also for doing all kinds of research in Alzheimer's disease. Now, among the dozens of studies that we do here, just a few examples of some of the treatment studies we are doing. One of them is known as the MIND study. Believe it or not, MIND is actually using a nicotine patch to see if nicotine can actually improve people who have a diagnosis of mild cognitive impairment. Now, that doesn't mean you go back and start smoking tonight, but actually there's some evidence that nicotine can activate certain parts of the brain which are impaired in Alzheimer's disease, and they help with memory. So it remains to be seen that if nicotine is given through a patch, skin patch, does it actually improve people with MCI and their memory? And the study is still ongoing. The other example is the Connectome study. Connectome is a brain imaging study where we're trying to find how different networks and parts of the brain communicate with each other and how they get impaired as we develop Alzheimer's disease. So this is a very special brain imaging study. Dr. Bendlin is here, and Dr. Sterling Johnson, and many others are involved with this study. It's a national study. We are one of the prominent sites for this study, and we are still enrolling people for that study. Another study is the Ambition study. Again, Dr. Bendlin is the principal investigator for this study. What this study is doing is actually giving medication or forms where microbes are given to people through oral route. Now, there's increasing evidence through Dr. Bendlin's work and other people's work that bacteria and microbes in our gut, we all have them from the moment we are born, they may have some role in Alzheimer's disease. And perhaps that can be used as a form of treatment for Alzheimer's disease. So what Dr. Bendlin and our group is doing, they're giving the more effective forms of microbes through a capsule that go into gut with the hope that they will improve Alzheimer's disease and perhaps reduce the risk and progression of MCI to Alzheimer's disease. So they're using participants with and without memory complaints. So this study is ongoing. Very exciting results from the center. Let's see if it turns out to be true. And then we have other studies. One of them is the BRAVE study. I know Dr. Carlson is here somewhere, right there at the back. Dr. Carlson is doing a very special form of fish oil and trying to find if fish oil can actually reduce the risk of Alzheimer's disease in people who are at risk for the disease. Now, this study is right now being done in only veterans, but if the initial result at the VA hospital here, but if the initial results are promising, I bet you that Dr. Carlson will conduct a much larger national study to see if a very special form of fish oil could be used to prevent Alzheimer's disease. So these are all just some few examples of some very exciting, innovative studies that are being done right here at the ADRC. Now, as you know, it's a large program. We have over 33 scientists that are involved. Of course, the phone will go off right now, right? Won't it? And I bet you it is a, oh, well, I had put my phone off. I don't know how it got on. Anyway, there's only so much of technology I can deal with. Yeah. Can't even put my phone off. How about that? Anyway, so there are a lot of ways that you can be a part of the center, and you can go on the website and actually find out how you can be a part of a center. And finally, Carol mentioned to us the initiative to end Alzheimer's disease that the University of Wisconsin and the medical school here launched about 3 or 4 years ago. And the role of the initiative is to find a cure and win this disease. And what the initiative is doing is trying to raise funds for research for Alzheimer's disease. Some of you may not know that every dollar that the NIH gives us, they cut us short by about 40 cents. So we have to raise 40% of additional funds to complete a study. And the only way we can do it is through fundraising and donation from people like you and across the state and nationally as well. And every dollar that you give us is utilized by the ADRC in our program to promote the program and also to do research. So it's, you don't have to think of doing $1,000 or even $5, $10, whatever one can afford goes towards supporting our center and the research. And these small amounts of donations have really helped us launch very high-risk, innovative studies that NIH is reluctant to fund because they want to be in a safer area. But the high-risk studies are the ones that really lead to breakthroughs in any research, including Alzheimer's disease. So Initiative to End Alzheimer's is that initiative to fundraise for Alzheimer's research right here at the ADRC and University of Wisconsin. And finally, it's my great pleasure to invite Dr. Carey Gleason, who will introduce our keynote speaker. Dr. Gleason is the Associate Professor of Madison in the Department of Medicine, and she is a prominent, nationally prominent, internationally renowned scientist in Alzheimer's research. Her real focus of research is in outreach and also community-based research and how to find how Alzheimer's disease differs between races and ethnicities. Our own research is focused on Native Indians and also American Indians and also on African American race. So it is a true pleasure and a privilege to invite Dr. Gleason to introduce our keynote speaker. Dr. Gleason.
Thank you. Everyone can hear me okay? Good. It's so nice to see so many familiar faces and to be together again for this fall lecture. So as Dr. Asthana mentioned, it's my job to introduce our keynote speaker, and it's my great privilege to introduce Dr. Neil Henderson. Dr. Henderson is a professor in the Department of Family Medicine and Biobehavioral Health at the University of Minnesota Medical School's Duluth campus. He earned his PhD in medical anthropology from the University of Florida, after which he worked as a dementia and diabetes researcher at the University of South Florida and the University of Oklahoma. Dr. Henderson was recruited to the University of Minnesota School of Medicine's Duluth campus, where he is the executive director of the Memory Keepers Medical Discovery Team on Health Disparities. The team is focused on ending American Indian and rural health disparities. So Dr. Henderson is also a registered member of Oklahoma Choctaw and has dedicated his career to understanding dementia and diabetes in American Indian people. He's also led initiatives to develop culturally sensitive community health interventions in education. So many of you have probably heard of the Savvy Caregiver program. Dr. Henderson led the initiative to adapt this for Indian country and is nationally recognized as a leader in caregiver training. So on a more personal note, for those of you who, I'm sure this is true for many fields, but there are sort of these glitterati and sort of heroes in any field. So there are heroes in science field as well. And what makes a hero for me in terms of their science work is their writing is clear, it's innovative, it's interesting, it draws a person in, and it adds something new to the field. So when I started working with the Oneida Nation of Wisconsin, I looked to the literature to understand the impact of dementia in Indian country, and so I was reading Dr. Henderson's papers. So he's a science hero of mine. And when I was traveling, I was in the airport, and he and his wife were traveling through, I think they were in a security line, and I ran up to him and said, "Dr. Henderson, I'm a huge fan." So he was kind enough to actually talk to me, and we exchanged cards, and it has led to a very fruitful and rewarding side for my research area. And I've learned so much from Dr. Henderson, and I appreciate that. So he is indeed one of my science heroes, and it is a great pleasure to welcome him to Wisconsin and welcome him to the podium. He'll be talking with us about diabetes and dementia among American Indians, connections and prevention. So I also invite you to approach him afterwards with your own science hero story. So thank you.
Alito, Chima Chukma, Saho Chefa Joniel Henderson, Chata Sia Hoke. Now, I just said hello to you in the Choctaw language and hoped everyone was doing well. I also offered a thank you to elders in that. And yet, I'm speaking to people here that themselves actually know some Choctaw. Everyone in this room knows at least 3 Choctaw words. You'll know one of the primary colors. You will know how to say all of humankind. And you will know how to say things are going well. And so, to backtrack on that, the primary color is Hama. The word for all of people is Okla. Let me ask you to sort of put those together, Oklahoma. It is a compound Choctaw word that actually means people red. You also, however, know how to say things are doing well. And I would bet that everyone in here, even today, has said this word sometime. And I'm going to tell you about a photograph I saw of a approximately 170-year-old letter from Andrew Jackson, who swept the southeastern Indians out of their homes over to Indian Territory. And while in the Choctaw Nation area, which is the Mississippi River Valley area originally, he picked up a word, and in a memo back to Washington, D.C., said things are H-O-K-E, and in Choctaw it's pronounced Hoke. Linguists will have debates about whether it actually came from that or not, but there is that connection And today, we say, how are you doing? And the response would be, okay. There's a H in aspiration there at the beginning of it. I want to also, before going on, to put on one of these get-togethers like this. It takes a huge amount of effort and a large number of people. And so I really do thank Dr. Asthana for his work here and for inviting me here. Dr. Carey Gleason, who has been so generous with the research work that she's done through the ADC here with Oneida Nation. And I came up to University of Minnesota about 3 years ago from Oklahoma. And Oklahoma right now has 39 federally recognized tribes. Minnesota has 11, but I didn't know anybody there. And so one of my starting points was to get to know tribal communities, and that is a thing that just takes some time. And Carey Gleason and I had become aware of each other now being neighbors. And so she very generously introduced me and members of my team to the work that she is doing with the Oneida Nation and the people of the Oneida Nation who have been so responsive. And so I really do appreciate that greatly. And last, like I said, there's many people, but Eva Davenport has been taxiing me around. But before that, we developed a kind of email deep relationship. So here I am, and I'm glad to be here. And I want to learn how to make this go. There we go. Okay. So by way of preview, I want to discuss tonight dementia and diabetes, but mainly from a prevention angle. Research today is showing very clearly that signs of the pathology of dementia shows up earlier in life and pre-symptomatically. And in some ways, diabetes can sneak up on people too, and they develop metabolic syndrome or the insulin resistance, which can then convert to actual type 2 diabetes. So early prevention is going to be a key here. Also, because we typically think about the effects of diabetes in terms of organ damage, with regard to amputations, with regard to kidneys, with regard to vision loss, it sort of directs us away from thinking about the conditions that are causing those organs to fail. Much of that is happening via the capillaries in the brain as well. And so there is a real connection there. And I would want to end with some things to do. It's one thing, I think all of us researchers are really good at dissecting things into little tiny pieces. And then when it gets to what do we do about that, it's a little more difficult. So I think we, however, have some things that can help us out. But first, please pay attention to this slide, if you would, and that just is to say that just because someone has type 2 diabetes does not mean that they will get dementia. Okay? It's a risk factor. More importantly, if it is managed well, then that can be neuroprotective all by itself. But managing it well does have to take place. We think that a lot of diabetes effects might be found in vascular lesions in the brain, still producing confusion and ultimately memory loss. But good management of it might mean that when people ask the question, so what starts this thing? What can we do about it? It may well be that we are, even right now, staring right in the face a means of prevention. If much of diabetes was either prevented or reduced, the idea is that a lot of downstream dementia and some significant proportion of the amount of dementia might not ever happen or might be stopped. So I'm asking us tonight to think about not just diabetes as an induced condition that is mainly a lifestyle effect that we in a way can kind of do to ourselves or we can kind of prevent, but think about some proportion of dementia related to diabetes as an induced condition too. We're saying, how does this happen? We may be doing it to ourselves to some degree. Well, here's how this works. And this is the results from many decades of longitudinal studies on what causes health and what's associated with it. Very broadly, the more money a person has, the more health that they have. The less money they have, the less health that they have. Then, with less health, you get more toxic stress. And keeping in mind, we're all stressed. If we're conscious, we're stressed. But toxic stress is a different kind of stress, and that is associated with less health. With less health, we self-treat with comfort food. High carbs, high starches, for example. It's called comfort food for a reason, and that produces less health. A lot of that comfort food ends up being high sugar foods, junk foods, and that's associated now specifically with type 2 diabetes. And we're saying the more diabetes, more dementia. So notice what's happened here. We've gone from a non-biological thing, a dollar bill, to a highly biological pair of conditions. That would be mediated largely through this toxic stress piece, probably. So, we're going to start talking about Alzheimer's disease and dementia and diabetes in the grocery store. Right down the street from us here, I bet you there's a grocery store that is just as filled as that one, and there's probably 30 or 40 of them across Madison. But we live, just happens to be, that we live in one of the places on the earth with a hypercaloric environment. That means we are just surrounded by constant amounts of food. And some of that food is high sugar food, and some of it is alcohol food. And if we know that we should avoid these things, why don't we? One thing is that identifying where these sugars are in that grocery store is made difficult on purpose. These are marshmallows. If you see down in the bottom left-hand corner, right in here, naturally fat-free, and it's gluten-free. For a consumer, it's almost like a health food. And apparently, it's not illegal to market that way. And in fact, it's called neuromarketing. I didn't make this up. This is an actual specialty within marketing. And the point of it is that the blue part, this blue cerebral cortex portion up here, some rational thinking takes place there, but we're going to skip that and go right into the limbic system here, our emotional kind of centers, the emotional triggers. And there is a particular way of doing that. So I take this from a guy named Hank Cardello, who was an executive with the National Grocery Association. And in it, he reveals some of the industry secrets, and one of them is that we might feel like the grocery store down the street is there to feed us and keep us well and healthy and happy. What he says is that it's down there to just sell products, and if you will buy it, we will sell it. And so one of the things that is done is at the entrance to grocery stores, the junk food, the pastries, anyway, the pastries and breads and pies and things like that are put right up there. They found most people hit the grocery store and they're hungry. That's the first thing you encounter. And then you try to leave, and you're further bombarded by all of these easily picked up items that are high sugar things. But one of the things that caught my attention the most is number 2, the kids' pester shelf. It has to do with the shelf layout strategy. And so people did anthropometric measurements of children from age 5 to 10. And they did not want to know just how high the top of their head was. They were measured for their eye level. And they placed the product at that level. And this is what that layout looks like. Those are for the adults. And right there, you get that. The bottom shelf is stuff few people would be picking up. So does that really happen? So I don't know if you can read from the back, but that's Fruit Loops and Kicks. And notice there are all these bright colors, cartoon images, glittery-looking, sugary content. And so the kid picks it up. They can reach it. They can see it. But then they pester mom until she finally gives in. And they take it home and down it. But let me ask you to think about this. Think about the fact that these neuromarketers, what if we think about it as they're exploiting a child's immature brain? And apparently that's just okay. With diabetes, there's a big S on purpose and underlined, because like with most things, it's not just one thing. And there's a number of items in print up here. Let me just run through these. The presence and amount of diabetes increases with age. You'll see this over half 55, and then as the age goes up, it continues to increase. That makes those people more likely to die earlier than someone without type 2 diabetes. Due to nerve problems in the extremities, and in this case particularly in the feet, is a higher risk for falls, which could result in surgeries, which could result in a mortality. Contributes to incontinence, and is a predictor for institutional long-term care. So it is something that is to be avoided. And yet, in the American Indian population, there is almost 3 times as much type 2 diabetes as in the majority population. So Indian country hasn't been sitting still doing nothing about it. You see here nearly 1 out of 6 American Indian and Alaska Natives has diabetes. And over on the right side, reversing diabetes, youth obesity and diabetes rates have not increased, which is another way of saying they haven't gone down, but they haven't increased. And yet buried up in this block here shows an increase from 2006 to 2013 in adult type 2 diabetes, recalling now that the frequency of it increases as we age. So there still is work to do. The amount is not evenly distributed across all populations, the amount of diabetes. American Indian, Alaska Natives are this bottom line, that big long line. The top one is the non-Hispanic white group. But so you see, this is affecting different populations in different ways. The yellow part of this pie chart is American Indian, Alaska Natives. Now, this isn't the amount of diabetes that's out there, it's death from it. The opposing purplish color up there are Pacific Islanders. So what we're doing, if you think about this, these are in reference to indigenous people, even geographically separated. So the diabetes business is an important thing in general, as well as with regard to brain health. Causes, plural, again, of dementia. And this is hugely complex, so I don't think that I can reel out all of the causes. But one of the things that has occurred that makes both of these conditions particularly important in today's world is that So it is literally true that there has never been a time in which so many people have lived for so long. And this is not ancient history. It's not something in the distant future. This is happening right this second, right this minute. As I'm getting older, every minute I'm part of this. So some of these matters that we're dealing with really are first-time issues to address. This is the Alzheimer's disease-related disorders, ADRD, increase from 2010 to 2060. And I think everyone here probably is very familiar with this projected dramatic increase in the amount of ADRD. And this is the demographics of the increase in aging in American Indian populations. So what we're seeing here in this population as well is people living longer and longer and longer. And another way of saying that is people are living more frequently into the ages of greatest risk for showing symptoms of some kind of dementia. And American Indian people, not so long ago, died 17 years before a corresponding white person. That has been shortened to 3 years now. Premature mortality in that population. And in some portions of North America, that is still really bad at around age 55 of premature death. So it's more work still to go there. Now this, I think, is an important slide because it is one of these answers to the question of what causes Alzheimer's disease, which of course we all want to know. And it is not absolutely known still at this point. But one of the things you see up here in the center of it is the image of a brain and beta amyloid and phosphorylated tau. But aging and genetics are remain the two absolutely known contributors to dementia. But we are, I think, in this room and elsewhere, experiencing a paradigm shift in the science of trying to understand Alzheimer's disease and other dementias. And that shift is away from exclusively looking at beta amyloid and tau. Those, by the way, remain crucially important. We don't mean that we've left those, but we're adding to that a large array of other factors that can potentially be contributing to this person's development of dementia, ranging from, and by the way, here's gut microbiome, which anticipates Dr. Bendlin's talk. Here we have diet and exercise. That's certainly related to metabolic health, avoidance of diabetes. And here's vascular risk factors up here. A number of possible inputs. I might even say probable inputs. It's just that we don't yet have the science to know what is the proportion of that contribution. But just be advised that because it doesn't say the word diabetes, that it's not buried in here in this diet and exercise portion. Let me have you look at this top part, the green, cognitive impairment. Stroke contributes to it, vascular dementia contributes to it, Alzheimer's disease contributes to it. Age, diabetes, diabetes, diabetes. Those are the only two that cut across all of those boxes. So how do these things and their different locations in the body, how do they actually get together? Let's start here. The brain runs mainly on glucose, and quite a bit of it, but it has to have the right amount. So we can overload it by ingesting sugars and carbs that become sugars. We can overload it, and then we get into an imbalance called hyperglycemia, too much glucose, too much blood sugar in its concentration. And it has causes multiple problems, but it does include dysfunctions and possible long-term damage. So here's the big web, and I'm just going to focus on a portion of it. On this left side, here in the middle, these green bubbles, says cognitive impairment, vascular dementia, Alzheimer's disease. Notice that all of those boxes have arrows all pointing there, all of them. So over on the right side, okay, I'm looking at it this way now. On the left side, this grouping over here, lifestyle factors, that includes mood disorders. There's the aging and genes. On this far right side is hypertension and stroke from that. And here is insulin dysregulation and abnormal ability to use it, leaving a high concentration of glucose in the blood, which of course still goes to the brain. At the bottom right here in the purpley pink box is hypoglycemia, too little blood sugar. And so if you don't have enough blood sugar, the brain just can't work normally, but that's fairly fixable by taking in some more energy. But at the top, just a couple of things. Here's hyperglycemia, the blood sugar is too high. And we're going to focus just on this one portion here that reads endothelial dysfunction. And that refers to the cells that line the interior and exterior of a capillary in the brain. Capillary being one of these microscopic sized blood vessels that is about the width of one red cell, so very tiny. In spite of all of that, there is a complex architecture of multiple anatomy all up and down these capillaries that are important for the correct amount of glucose getting into the brain. So I think I have two of these. So this is one. This is a brain capillary, and what I want to point out in the middle here, it's like you've cut it like you would cut a garden hose, and now we're looking down the inside of it. That would be the blood flow that's in it, the blood cells. And this yellow representation is a cell called an endothelium, but there's thousands of them winding all throughout. There are other cells like this green one called a parasite. It's also helping to tend the integrity of that blood vessel. This is, as you see up here, where the blood-brain barrier mainly occurs. Well, what happens is that the glucose that's in this blood has to be transported out of it through the walls of this blood vessel, of this capillary, to get into the extracellular part of brain tissue so it can be used to let the brain operate. There are really fantastic chemistries that occur that transport that glucose out of the bloodstream and through the walls out here where it can be used. And so here's a brain cell over here to the far right. Now, and there are waste products that have to go back into it the other way and transport it out, clear out of the brain. So imagine that we have this high concentration of glucose in our blood, head to toe. But right now we're in the brain. And the ability of these endothelial cells to operate correctly and for the signaling systems to transport glucose out into its use in the brain. If that is damaged, then the brain is not able to get a sufficient amount of nutrients, of glucose, of oxygen even. And so this is the place where the diabetes dementia connection can occur. If this goes on without sufficient regulation of glucose, that is, the person is not managing this well, that's where the risk comes in for permanent damage to this neuron and possibly even death of that neuron, which will not replicate. So that's the location, but here's why. Forget about the diagram at the top. The bottom words summarize the effects on the brain. Right here on the left side, reduced cerebral blood flow. So it can't get even enough nutrients in O2 and glucose there, and it produces hypoxia because insufficient oxygen amounts. Blood-brain barrier dysfunction means that the balance that is physiologically tried to be achieved on the amount of glucose going in and out is not what it needs to be. The dynamics of all of this anatomy up here means the cells may die. And the reduced clearance allows for protein accumulation, all because of exposure to hyperglycemic conditions inside these blood vessels. So let's apply this now to American Indian people. Not a whole lot is known, by the way, but one of the earliest studies by a neurologist named Hugh Hendry looked at Manitoba, Canada, here's Winnipeg, and he compared a group of native people, native Cree people, to people in Winnipeg, Canada. And what he found was this, that the title, by the way, was one of the best titles I've ever seen in a science journal, and it was just simply Alzheimer's Rare in Cree. If they were all that straightforward and simple, we'd be very happy. So here's the numbers. Notice the big difference in the native Cree population, 0.5 versus 3.5. And now this is in the early 90s, so there was still a lot to learn. This was not, we would certainly not think of it as having an absolute diagnosis, but some indicators of it. Then when he changed it from Alzheimer's disease to dementia terms, more broadly cognitive impairment, then it happened in this case to equal what was in Winnipeg. But the Alzheimer's type he considered to be rare. Staying in Canada, but shifting from the early 1990s to the mid-2000s, is work done by my associate director, Dr. Kristen Jacklin, and her team, that shows in southern Ontario the following. If you look over here on the left, you can see the prevalence is around 34% in First Nations people in southern Canada. There's earlier signs of onset. There are more men than women, and the rate of developing the condition is faster, and if we knew why all of these things were happening, we would be very happy. But these currently constitute research questions for us about Native people. Now, shifting to more work in the early 90s by a guy named Roger Rosenberg, and he's working with Oklahoma Cherokee, which is this top reddish color, northeastern part of Oklahoma. And his findings were the following. Those are the criteria, the diagnostic or screening criteria that were used. And so what he found was the following here. That is that when we, if we're going to do genetic work or molecular work or medical work with American Indian people, and I think actually most people, but certainly here, we need to understand a little bit about genetic admixture so that some people have a very high genetic load of North American indigenous genes, whereas other people are tribal members, but they have a lower amount of indigenous genes. And so for these kinds of studies, that can make a difference. And what he's saying in this last bullet is the following. If a person has a high degree of indigenous genes, then the development of Alzheimer's type dementia is lower. So it suggests there may be some protection of some kind in population genetics for Alzheimer's among these. When ApoE4 was put into it, it didn't make any difference with regard to developing Alzheimer's or not. Next, in Oklahoma, in the bottom corner here in the brown is the Choctaw Nation of Oklahoma. And nearly I forgot to put this in here. So, in hospitals, phlebotomists that go around drawing blood are often called vampires or bats. So that is the word for Choctaw bat, which is one of our good friends, CHR, is doing a blood draw there. And what I found was this with regard to the frequency of E4. The tall right column is the frequency for the white population. This short one over here is the frequency of E4 allele in people that have the highest genetic load of indigenous genes and were over 55 years of age. So independently, Roger Rosenberg and I found a similar kind of effect with regard to E4. And what happens as the frequency goes up here is more and more European or Caucasian genes were added into those individuals, and the presence of E4 went up. So we found an inverse relationship also, and we found that if E4 was present, it didn't matter. And so the resilience to that is an interesting question that people are exploring also. But one of the important things here is that Cherokee people are Iroquoian speakers, and they are in, at contact time, we're in the eastern Carolinas, and that's where the federal reservation is today. Choctaw speakers are Muskogean speakers, and all those southeastern Seminoles, Chickasaws, Choctaws, Creeks are all Muskogean speakers. So what it means is the people we call the Cherokee were formerly Iroquoian people, migrated for some reason, some portion migrated for some reason into the Carolinas. Now we're swept out and we find them in Indian Territory. So the point here is that Rosenberg and I had similar kinds of findings from tribes that came from two very different geographic areas. It just makes us wonder to what degree is that a representative profile? Does that apply to other tribes or all native people? We certainly don't know right now. But the finding is this: the more that there is an American Indian genetic load, the less presence of E4 might suggest that there is some kind of protective benefit operating somehow. So if there's not much of this going on, is that reflected linguistically? And so I would ask older people for what would you call this, or what did you call this? Was there a name for this confusional state we call dementia? And they would run through these kind of candidates almost universally. So Tasimbo, Okpalat Chaha, which is crazy, and then Lupia Chukma Ku. This Lupia's brain, the Chukma is good, Ku is not. But then they would kind of settle on this one, and it is Imanikvila Kania, which means the intellect is no longer there, but it doesn't mean it's gone, it means it's displaced. The intellect is still present, but we can't access that at this point. So there is cultural features with American Indian people. We've talked about some of the biology now, but cultural features of this includes the following here, and that is Pokney is the Choctaw word for grandmother. So this woman, Mrs. Maytubby, is an older Choctaw woman, is what that says under there. Maytubby is a Choctaw name that comes from the native language and then is drastically altered into English, but it's not Tubby, it's actually Ubby, which means killer. So watch out. Well, the granddaughter told me as we were there visiting with her Pokney there that she sees things we don't. And think how different that is if someone had said, she sees things that aren't there. She's hallucinating, she's got a brain disease, this is really strange, and just let her chatter on. That's not how it was interpreted. It was interpreted as Mrs. Maytubby literally looking into the other side before she died and describing to people, the lucid people around in the room, the names of people that had walked on before favorite horses, favorite dogs, talked about vegetable garden. And this wasn't just one time, that she would do this with some frequency. And so, in a social cultural way, it became a little bit of a special honor to have this happening in our presence. We're seeing that there really is another side, that there are recognizable people, even recognizable animals, and that she was talking with them in preparation for going there. In southwestern Oklahoma, with end-of-life matters and southwestern tribes, Plains Indians people, they talk about knowing the person was going to imminently go to the other side when they began to speak a little more lucidly, and they would talk about seeing their relatives coming to get them. And that's when the family, then, the lucid family, would start singing prayer songs. My Pokney, Choctaw, died in a nursing home bed with my mother and her sister, who got into the bed with her, and as they said, sang her over to the other side. So one kind of brain disease caused by many possibly different things, and interpreted and responded to in very different ways. Researchers, like all of us, and participants benefit from understanding and being able to better communicate with people in different population segments by being open to these varieties of interpretation that go along with heartfelt feelings. So I want to thank you all for your time, and I appreciate the work that you do as caregivers and the professionals here and the ADC. You all right here in this room in Madison really are among academic research stars, honest to goodness, and just because they're local doesn't mean they're not stars. They're national and international stars, and I appreciate getting to be here. Okay.
Thank you. Thank you so much, Dr. Henderson. Okay, what does that mean again? Okay? What is the word okay? Okay? Okay. Well, that was fascinating. I learned all kinds of things that I never even considered before. I was particularly pleased to know that Dr. Henderson was going to be speaking with us tonight on a personal level, because when I saw that he was from the University of Minnesota in Duluth, well, I'm from Superior. And so we immediately connected, and I said, was it snowing when you left? Thinking I was going to tease him a little bit, and he said, well, actually it was. There was snow flurries in the air when he went to the airport this morning in Duluth, and I said, yeah, I can believe that. But anyway, welcome to Madison, and it's wonderful to have a hometown person with us. And now on to our next speaker, who is Dr. Barbara Bendlin. And Dr. Bendlin is an associate professor of medicine and geriatrics and an investigator with the Wisconsin Alzheimer's Disease Research Center. Her lab examines the interplay of factors that contribute to healthy or diseased brain aging. She also studies how modifiable factors, those lifestyle factors, such as sleep, diet, and then microbial influences, that's that gut health we've been talking about, or midlife metabolic disorders may contribute to or actually protect against Alzheimer's disease. So please join me in welcoming Dr. Barbara Bendlin.
Thank you, Carol. And thank you, Dr. Henderson, for coming to join us and giving such a wonderful presentation. My name is Barb Bendlin. I'm an associate professor at the Department of Medicine, and I went to school in Winnipeg, Manitoba. So I'm just completing the circle of connections here. Oh, I'm sorry we didn't have time for these. These are great. Please tell us what to do. Oh, this is perfect. plant good things in your brain. And actually, I'm going to talk about this in my presentation. I'll be speaking on microbes and Alzheimer's disease. And I'm starting my presentation as well with just with a short story. We're interested in factors that can increase or decrease risk for dementia, and really trying to figure out, why do some people show healthy trajectories of aging and other people don't. And so I have an example of two individuals here. This woman on the left, does anybody know who this is? This is Pat Summit. She coached the Lady Vols basketball team, women's basketball team, and when she retired, she had the most wins of any coach in women's or men's collegiate basketball. And she was very successful. She retired when she was 59, though, when she was diagnosed with Alzheimer's disease, dementia. And she passed away 5 years later at the age of 64. On the other side, we have Dr. John Carey Gleason, who lived in the Lake of the Woods region. And he, as far as I know, is the person to live the oldest documented life up to the age of 124. So he lived in Minnesota, and he was a medicine man, and he was actively working on his trap line up until the age of 110. So here we have two people with very different trajectories of aging. And so Dr. Henderson talked about differences in genetics. That's certainly something that we know contributes. But we also know, for example, even among genetically identical twins who have the same genetics, that there can be differences in terms of their age of onset of Alzheimer's dementia, up to a documented 18 years difference where one twin develops the disease, Alzheimer's dementia, and the other one doesn't. So there are other factors that we believe play a role. One of the things that my lab and in collaboration with others on campus have been looking at is the contribution of gut microbiome. And this is something that you may have heard about in the media, but if not, that's fine. I'll tell you a little bit about what we're talking about. When we're talking about microbes, we're talking about very small organisms that are everywhere in our environment. And the ones that we're interested in live in and on our body. And this includes bacteria, viruses, fungi. And with regards to where they are on our body, this is a map of where they are on our skin. The majority of them actually live in our gut, where they play very important roles in health. How many of these little microorganisms that we can't see are in and on our body? Actually, more of them than there are human cells. So there are about 38 trillion microbial cells in and on our body versus only 30 trillion human cells. They're very, very small, so by weight, we are still more human. With regards to genetics, if we're talking about humans, we have 25,000 genes or less. With regard to microbes, because there are so many different kinds, the genetics is much, much higher, so 3.3 million genes. And if you think, we're not really in the practice of changing human genetics, but we might be able to modify the microbes that live in and on our bodies. So if we're interested in the microbes that are in our gut, how do we study them? The main way that we do this is actually to look at stool samples, because what's in our gut comes out, and then we can actually use those stool samples to see what is in the gut. So we use a type of genetic sequencing to figure out what is there, how much is there, how many different kinds of things are there. Another really interesting tool we have for figuring out what microbes do in health is to study animals, and in this case, mice. Mice that have been kept germ-free, so they've never been exposed to any germs. And then what we can do is we can introduce microbes and see what happens to their health. So let me just tell you a quick story about some research done here on campus, well, actually done by an investigator here on campus, Dr. Federico Rey in the Department of Bacteriology, and he was studying obesity. And he has a colony of these mice that are kept germ-free. They've never been exposed to any microbes. And if you feed these mice a high-fat and high-sugar diet, they do not gain weight. Okay, that doesn't mean that we should all become germ-free and live in this bubble, because it's not very enriching. But then what they did was really, really interesting. So they wanted to figure out how human microbes would affect obesity. So what they did was they collected stool samples from twins where one twin was normal weight and the other twin was overweight. Now what you can do is take those stool samples and give them to the mice so that they have a human gut microbiome. If you give these mice a stool sample from a normal weight twin and feed them a high-fat, high-sugar diet, they do not gain weight. If you take a stool sample from the overweight twin and give it to these mice, and then feed them a high-fat, high-sugar diet, the mice gain weight, and they also develop basically type 2 diabetes. So there's something in those gut microbes that's really influencing the health of the mice and presumably of the humans as well. So we know that these microbes have been studied in the context of a lot of diseases that affect the gut. So things like Crohn's and colitis, metabolic syndrome, type 2 diabetes. But now there's more research looking at the effects of these microbes on the brain. And that is because the gut is actually well connected with the brain through something called the vagus nerve. And also these microbes in the gut produce signaling molecules that send messages to the brain. And this is a very, very close connection. And this is a list of just some of the brain conditions and brain diseases that have been associated with gut microbiome. And probably out of all of these, the one that's been most studied in terms of the context of neurodegenerative disease is Parkinson's. In fact, there's a pretty interesting emerging story about Parkinson's, because some of the brain pathology that you see in Parkinson's, the abnormal accumulation of alpha-synuclein pathology, is also found in the gut. So there's a theory that the disease might even start in the gut. With regard to Alzheimer's disease, we're looking at a different kind of pathology. We're looking at beta amyloid plaques and neurofibrillary tangles. And what's super interesting is if you take a mouse that overproduces beta amyloid and you keep it germ-free, it does not develop as many amyloid plaques in the brain. If you then put microbes in the brain of those animals, the amyloid comes in to actually sequester the microbe. And there's a theory that potentially microbes or microbial products getting into the brain could actually instigate this accumulation of the beta amyloid plaque. Now, this is a theory in Alzheimer's disease. This is something that people are working on and needs more study. So we at Wisconsin have been looking at gut microbiome differences in people with and without Alzheimer's dementia. So these are research participants that come in and participate in all of the procedures that we ask them to do. They're very generous with their time. Some participants even undergo a lumbar puncture to provide cerebral spinal fluid, which tells us about pathology in the brain. And then the easy part is providing the stool sample. So that part is done at home. And what we were interested in was seeing if in Alzheimer's disease there's differences in the gut microbiome. And what we did was to see what was there and how much was there, and also how diverse was the gut microbiome. So if you think kind of a healthy ecosystem, there's lots of different kinds of plants and animals, that would be considered healthy because it's very diverse, versus a lack of diversity or monoculture. That's what you might see in a disease state. And this work was carried out by Nick Vogt, who's a student at the university, an MD PhD student. And what he found was that actually in Alzheimer's disease, there's decreased diversity, so not as many things in the gut. But of course, once people develop dementia, their diet changes. They're also on medications that could affect the gut. So what Nick did was to actually look at that cerebral spinal fluid and look in people who did not have dementia. And what he found was that even in those individuals, the bacteria in the gut that he was studying were related to the pathology that you see in the brain. So there is this link between the gut bacteria and what you see in the brain even before people develop dementia. And the studies in the mice are ongoing. So if we know that the gut influences the brain, how do we change what's going on in our gut? So there's a number of different things that affect your gut microbiome. We get most of our microbes when we're born from our mother. If we have a normal, or if we have a vaginal birth, then we will have the same microbes as our mother. Who we live with actually influences our gut microbes. In fact, your gut microbes start to look more like your spouse than even your siblings over time. And that's actually more so the case if you get along with your spouse. We know that antibiotics can affect gut microbiome, and diet, of course, is one that significantly affects gut microbiome. This is very briefly just an interesting study looking at immigration, and these were native Cairn and Hmong individuals who lived in Thailand. They were studied in their native Thailand. They actually had very diverse gut microbiomes. When they moved to the United States, they lost diversity, and their offspring, so their children and the next generation, also lost gut diversity. So there was a westernization of the gut microbiome. That was also associated with greater obesity. In contrast, from the American Gut Project, they compared individuals, they looked at gut microbiomes, and they compared individuals who ate 30 or more different plants per week to people who ate 10 or less different plants per week. And eating 30 or greater different kinds of plants per week was associated with greater diversity. The last little bit that I wanted to talk about, if there was a more radical change of the gut microbiome, that would involve a fecal microbiota transplant. And fecal microbiota transplant or stool transplant means actually taking stool from a healthy donor and using it to increase diversity in someone who has reduced diversity. And some of you may have heard about this in the context of C. difficile, a hospital-acquired infection. Actually, at our UW hospital, they use stool transplant to treat C. difficile if it doesn't respond to antibiotics. So you can take someone who has a depleted diversity of microbes and use a stool transplant to increase diversity. And so there's now a number of studies around the country, clinical trials, that are testing stool transplant for a number of different diseases. And in Wisconsin, we have the first feasibility study to test this among individuals with Alzheimer's dementia. So in summary, in that short amount of time we had, we'll have questions at the end, but what is it that you need to know about microbes? So microbes have been linked to a number of health conditions. They're starting to be linked to brain diseases, and it needs a lot more research to figure out what are those mechanisms. But we believe that this could be potentially another opportunity for intervention. All right, thank you.
Thank you so much, Dr. Bendlin. If I'm hearing you correctly, when you talk about those 30 plants and the different varieties, that just the basic first step for us is that rainbow of colors on our plate in both vegetables and fruits. And if we start there, we might be on the right track. Thank you so much. And now on to our next speaker, who is Dr. Nathaniel Chin. Dr. Chin is an assistant professor of medicine and geriatrics and a memory care specialist at UW Health. As an investigator in the Wisconsin Alzheimer's Disease Research Center, Dr. Chin develops and supports programs that help patients and families navigate a dementia diagnosis and disease progression. He is also the host of the podcast Dementia Matters, which introduces research and caregiver strategies to a general audience. So I consider him a colleague in broadcasting, but you're here tonight as a researcher and clinician. So please join me in welcoming Dr. Nathaniel Chin.
Thank you, Carol. I'm flattered by that comment. Thank you. And thank you all for being here today. I have the privilege of ending our lectures. And really, this topic, we at the Wisconsin Alzheimer's Disease Research Center wanted to take this opportunity to talk with you about this evolving definition of Alzheimer's disease, which I imagine many of you have heard about or read about in the news. Now, Alzheimer's disease is a complex disease, and so it's not surprising that how we define it is complicated. Important changes in how we view Alzheimer's disease were made in early 2018 that we think are going to have a major impact in how we conduct research and how we are conducting research. So for the next few minutes, I hope to shed light on this new definition and what it means to us. Now forgive me, I'm going to have to do a couple of changes here at the same time. Okay. All right. So when trying to define Alzheimer's disease, I think about it from two perspectives. Now for me, this is Alzheimer's disease. It is my father. It's the person. But of course, Alzheimer's disease isn't a person, though I believe those of us in the room that have a loved one affected would say that it feels that way. But another way of saying this is that we can view Alzheimer's disease for what it is on the outside. You can see the personality changes, the physical changes that people undergo, along with recognizing the changes in the memory and the thinking skills. Now, the second perspective of Alzheimer's disease is what it is on the inside, what we can't see in a living person, and that's the changes that occur in the brain. So Alzheimer's disease is actually an abnormal process that occurs in the brain, and it's caused by two abnormal proteins. Now, one protein forms outside of the brain cell, and we call that amyloid. And another protein forms inside of the brain cell, and we call that neurofibrillary tangle, and it's made up of a tau protein, so amyloid and tau. Now, these two proteins work together, and our brain cells die prematurely, leading to the symptoms that we see in people. Now, we can see these proteins under a microscope when looking at brain tissue, but that requires someone to die, and so it's not helpful when thinking about clinical trials. Now, in the doctor's office, we diagnose Alzheimer's disease based on what we see with the person, our patient. We look at the outside of this person to make this determination, and we use the history that's provided and the brain testing that we do in the clinic to first determine if someone has normal cognition or mild cognitive impairment or dementia. Now, the biggest difference between mild cognitive impairment and dementia is that someone with dementia has day-to-day functional impairments, such as a difficulty in managing medications, or in driving, or in handling their finances. And once we have that diagnosis, we use the details of the history, the pattern in the cognitive testing, the blood tests, and the brain imaging to help us determine if Alzheimer's disease is the primary cause. So there's no brain biopsy, and there's no looking for any of these abnormal proteins. Now, when I say that a person's clinical history helps us determine if Alzheimer's disease is the primary cause, what I'm referring to are the symptoms that a person has. So for instance, forgetting recent events or conversations, misplacing items, struggling to find a person's name. These are symptoms. But these symptoms vary from person to person because Alzheimer's disease can start in one place and then move in another. And this variation makes it difficult to predict a person's course of symptoms. Now, in general, Alzheimer's starts in the memory center, what we call the hippocampus. But from there, how it spreads can vary. And so if it spreads to the front part of the brain, then a person's going to have problems with problem solving and planning. But if the first part of the spread is to the language center, well, then a person's going to report word finding or difficulty understanding words. And in less common cases, the spread can go to the vision center, and then a person's going to report difficulty with judging distances or their depth perception. Now, the disease even spreads to this behavioral control center, which is why our loved one might feel agitated or even be socially inappropriate. So we see these symptoms not because the person chooses to have them, but because the disease is affecting that portion of the brain. So in research, Alzheimer's disease is based on what we see on the inside. And when we start with the brain itself, we see that there is shrinkage, and that shrinkage represents brain cell death. And in general, it can be the whole brain, but for some people, it's just that memory center, the hippocampus. So fortunately, we can identify these changes in living people through the use of MRI scans. But the problem with this is that brain shrinkage occurs later in the course of disease, and it's not unique to just Alzheimer's disease. So we need to go deeper and start looking at the cells that make up the brain tissue. And when we do this, we can identify those beta amyloid plaques and those neurofibrillary tangles. Now, these changes are unique to Alzheimer's disease, and they're important in correctly identifying the disease. So the good news is that with advancements in technology, we don't have to wait for brain tissue in order to find them. We can see them in living people through the use of studying spinal fluid and PET scans. And what I'm showing you here are 2 PET scans, with the one on the left showing us the presence of amyloid protein, and the one on the right showing us the presence of that tau protein. And that red and the yellow that you see, that's the presence of that protein. So with our improved ability to identify this disease in living people, specifically those 2 abnormal proteins of amyloid and tau, the research community came together to create this new framework. Now, this new framework is the evolving definition of Alzheimer's disease, and it represents a shift from clinical symptoms to a biological definition based on those proteins themselves. Now, making this change is not easy, and it's controversial at times, but it's meant to improve our research by getting everyone on the same page, using the same language, and studying the right people who actually have Alzheimer's disease. So here's the framework that the scientists and the Alzheimer's Association put together in 2018. Pretty straightforward. Pretty easy to understand, right? No, it's not. These brilliant scientists took a complex disease and they made it more complicated. And then they put it in this very intricate table. So I'm really not going to talk about it. They took another stab at it, and they gave us this picture. Much better. It's not a ton better, but it's something we can work with. So I want to spend a couple of minutes going over it. Let me see. So do I have a pointer? No. Oh, okay. Thank you, Michelle. All right. So what matters most in this figure is the yellow circle and the red circle. The yellow is amyloid, the red is tau. Now this green circle, well, this represents cognitive impairment. And you see that it's all-encompassing because Alzheimer's disease can cause it, but so can a lot of other diseases. And then we have the blue circle. That's brain cell death. So similar to the green one, lots of things can cause this, including Alzheimer's disease. So I want us to really ignore the green and the blue, and let's just focus on the yellow and the red. So this part that kind of looks like a moon, this is what we call having an Alzheimer's brain change. It just means that you have that amyloid protein. It is not Alzheimer's disease. It could be the beginning of the process, but it is not the same thing. In order to have Alzheimer's disease, you have to have amyloid and tau. So that's this overlap. So that's why they say Alzheimer's disease here. This is the key part of it. So this is an Alzheimer's change, but it's not the disease. This is Alzheimer's disease. Now this remaining red part, this means you have that tau protein that I was referring to, but it's not being caused by Alzheimer's disease. It's being caused by something else. A little bit clearer? Okay. No? Well, really, I just wanted to show that these brilliant scientists aren't brilliant at everything. So here's something that we created. So it's another one. What's that? A test at the end of it, yes. Even I couldn't understand that first table, so I'm not going to do that to you. But here's another way of looking at it, and now time is being considered. So we put this together. So we have amyloid forming in the brain, and while it's not Alzheimer's disease, it is the beginning of a process. And then tau tangles will form. And now amyloid and tau together mean that you have the research definition of Alzheimer's disease, regardless if you have symptoms. Now, at some point, these 2 proteins are going to lead to brain cell death, which we can pick up on with an MRI scan. So these are the same three things as the three circles in that last slide. And these are all research-driven. However, when these changes that we see start causing symptoms, well, now we've crossed this line and we're entering the clinical world. So a person with brain changes that has symptoms and performs poorly on brain testing, well, that's called mild cognitive impairment. And when those symptoms and the changes and the performance worsen, and a person also has day-to-day functional change, well, then that's what we call dementia. And so we call it dementia due to Alzheimer's disease because we believe the beginning process is due to that amyloid and tau. So you can start seeing how this research world is slowly making its way into the clinical world. So this evolving definition of Alzheimer's disease is important for researchers. By having the research definition rooted in disease biology, the amyloid and the tau, the research community now has a framework from which to conduct the right studies and communicate them to each other and to you, the general public. Now, having this framework allows for researchers in clinical trials to enroll the right people at the right time of their disease in the right study. So in the past, studies were being done or enrolling people based on clinical symptoms, which isn't always accurate if telling if someone has Alzheimer's disease or vascular disease or Lewy body disease. So in the past, we may not have been studying actual Alzheimer's disease. It could have been something else. So if we are to understand Alzheimer's disease and the interventions that could help it, then maybe we need to make sure that these participants actually have the disease. So moreover, when researchers are comparing studies or they're building studies off of each other, we need to make sure that we're talking about the same disease. Are we talking about a duck or are we talking about a rabbit? And that matters when we're talking about working with other centers across the country. So having the research definition be based on actual proteins in the brain opens up the possibility for specific treatment targets. In essence, addressing the cause of the fire and not just the flames. But lastly, we can study Alzheimer's disease by seeing how do these proteins interact with other aspects of our health. And what does that mean for our overall cognitive performance? Humans are complex because we're not isolated in vacuums or in petri dishes. We have other medical conditions, medications, lifestyle behaviors. So understanding how the brain performs in the real world with amyloid or with amyloid and tau is important in figuring out how to treat real people. Now currently, our drug trials are looking to reduce the buildup of amyloid or clear it out more effectively. And I know Dr. Asthana talked about that earlier. Now these studies haven't been entirely successful, but that means it may take more than just amyloid treatment. And so studies are being designed to address the tau protein too. But the thing is, by having amyloid and tau as the basis for this disease, we now have other targets we can look at. So for instance, what is happening between the development of amyloid and the development of tau? What is going on there? And is there a target for us to treat that? But in the meantime, while the drug trials are being created and they're underway, we need to be looking at things we can do now to prevent or modify this disease, things that are in our control. Now, this is something that our center is very active in and, dare I say, leading the charge. Now, my colleague, Dr. Lindsay Clark, recently published a study looking at how other health factors contribute to memory loss in the context of having high amyloid levels in your brain. So she looked at obesity, defined as having a larger waist circumference, and high blood pressure, defined as having a blood pressure greater than 140/90, to see what effect do these conditions have on brain performance, or really how did someone do on testing. Now, compared to people without any of these factors, high blood pressure did not worsen a person's cognitive performance. And the same was true for obesity. However, when you have obesity and a high level of amyloid in your brain, that tipped the scales. So there was this synergistic effect of having amyloid and one of those factors, and it led to a faster decline in cognitive performance. So the same is true for blood pressure as it was for obesity, but it was not true for depression and high amyloid levels. So there is something here, and it's important since we can actually treat obesity and treat high blood pressure. But we need to identify the right people to study and the right people to treat. which is why this evolving definition of Alzheimer's disease is so important and needed to further the research. So as was what was mentioned before, I have this podcast called Dementia Matters, which is launched by our own center. It's the only podcast of the 32 centers. And our aim is to provide education to the community about the latest in Alzheimer's disease research and caregiver issues. Now, this topic of the evolving definition of Alzheimer's disease is covered in a few of the interviews that I've recently done. So I encourage you to listen to it, but also to stay tuned, because the science is moving fast, and our center wants you to be informed, because hopefully that leads to making healthier decisions, but also we want to assure you that we are making progress thanks to you and your involvement in this work. Thank you.
Thank you so much, Dr. Chin. I'm glad that you posted where we could listen to your podcast. I will listen and learn. What a wonderful evening this has been, and a special thank you not only to Dr. Chin, but to all of our presenters this evening. So we could have a final round of applause just for the presenters. Yeah. Outstanding opportunity that we've been given this evening. And now we're going to move into the dialogue portion of the evening, which is the question and answer. And before that, I would like to remind you to complete that all-important evaluation form, that yellow sheet of paper. And then you have pens in your bags if you need one. And the staff from the Wisconsin Alzheimer's Disease Research Center will be collecting the forms as you exit. So just hand the form to one of them. And I want to thank all of you for coming this evening. You make this possible because this is a dialogue between what's happening at the university and the community at large. If we can have you pass your note cards now to the center of the aisle to the staff members, and they'll come down and collect the note cards with your questions on them, and we'll begin to field the questions. So I will now turn the podium over to Dr. Dorothy Edwards, a professor in the UW-Madison School of Education and the School of Medicine and Public Health. Dr. Edwards is the director of the UW's Collaborative Center for Health Equity. She's the co-principal investigator of the National All of Us Studies, which is here on the UW-Madison site. And she's the leader of the Wisconsin Alzheimer's Disease Research Center's outreach, retention, and education efforts. She's a very busy woman, and we're so happy to have her here with us this evening. Welcome, Dr. Edwards.
Thanks, Carol. Mostly I collect titles. They're gathering the note cards for your questions. I just want to say that I first met Dr. Henderson a long time ago, and I will tell you exactly when it was. We were at a meeting in North Carolina hosted by Dr. Peggy Dilworth-Anderson, who will be our Solomon Carter Fuller speaker this spring. But Dr. Dilworth-Anderson called together a group of people doing caregiving research because she was interested in the physiology, the physiological effects of long-term caregiving. She called together a group of people to put together a proposal which wasn't funded. It was way before its time. But I first heard Dr. Henderson's research and his description of how Choctaw elders define dementia differently really made an impact on me. I can't tell you the year because I don't remember the year, but I will tell you that while he was speaking, my beloved St. Louis Cardinals gave the World Series to the Boston Red Sox. So whatever year, bad year that was when Dr. Henderson was speaking. I think it was in 1987. But so his work has had a really major impact on my work and the work of many others. And I think you can see that despite differences in race and ethnicity, in environment, whether you live in Canada, the United States, that we all share common risks and also preventive factors for dementia. So in groups that have more risk, who have accelerated rates of Alzheimer's disease, sort of accelerated aging, we can actually see the processes that benefit everyone. And so I really want to thank him for his work. So I'm waiting for the questions. I get to decide which questions our panelists answer, but I'm going to call my panelists up here also. I also want to thank Dr. Bendlin, whose work on the microbiome is really, really important. I happened to be at a UW Madison day in Congress several years ago, and Dr. Bendlin spoke to the US Congress, one of the Science and Technology Committee actually, about her work on the microbiome. So she's a rock star in the microbiome world. So, lots of questions.
Actually, Dorothy, while you sort through those questions, I just also wanted to take an opportunity to thank Carol for emceeing our event, and she's been doing this for many years, so thank you.
Okay, I'm going to start with this one. I think this goes to Dr. Gleason. Are there some personality traits that affect the likelihood of developing Alzheimer's disease? Yes, actually.
How's that? Okay. Yeah, there are. There's been studies from a couple of different groups that looked at different personalities in terms of risk factor for dementia. So there were indeed links between people familiar with the big five personalities parameters. Neuroticism was one that was linked with the risk for dementia, and the term neuroticism doesn't necessarily refer to the old-fashioned term of neurotic, but it relates more to sort of a coping style of sort of anxiousness, perhaps, maybe some rigidity. But there have been, most likely, those are probably related to the behavioral outcomes of some of the personality features. But I also really like to make a sort of side comment here that we don't inadvertently blame people for getting dementia or for Alzheimer's disease dementia. The personality features or the personality variants exist in our culture for a reason. It's really important that we not then turn it back around and say to the person, you have dementia because of who you are. In fact, what I'd like to do is actually turn it the other direction and say, you have an opportunity to address this with modifiable risk factors. So if a person has factors or personality features that interfere with their ability to function in day-to-day life, if it prevents them from engaging in healthy behaviors, here's your opportunity to address that with sort of maybe it's a talk therapy or a medication that can help a person sort of get into a healthier groove. So hopefully that answered the question related to personality features, but it does indeed, there is a link there.
Thank you. So I've gotten 4 questions on gut microbiodiversity, Barb, and the effects of colonoscopy, and whether having a colonoscopy actually, because of the cleansing that has to be done before a colonoscopy, whether it actually affects your gut microbiome and are people actually at greater risk of Alzheimer's disease.
Right. So actually, the things that potentially disturb our gut microbiome, like even antibiotics or colonoscopy, it turns out that your gut microbiome bounces back. So even if we take a course of antibiotics, it changes and it decreases the amount of microbes and diversity, but it comes back almost to the same. In the case of antibiotics, not quite, but almost. And as far as I know with colonoscopy, it also, it bounces back. Yeah. So it won't be a cure.
Can you also speak to probiotics and how probiotics affect?
Yeah, so that's the other thing is if we could figure out what's missing in Alzheimer's, could we put it back with probiotics? And some people think that's something that we might be able to do. But again, when we take probiotics, it's just a very, very small drop in the bucket. So you have so many microbes there and so many different kinds. Usually when you're buying something off the shelf, it's only going to be 2 or 3 different microbes. And you take those, and then as soon as you stop taking them, they're pretty much gone. So it's hard to get them to colonize. So generally speaking, what's a little bit, maybe another idea would be to feed the ones that you have there. And then again, that gets back to we have a lot of different kinds of things there, and so we need to feed them different kinds of food. Some people also talk about prebiotics, it's things that microbes eat. Feed them to grow what you have there.
Thank you. Dr. Chin, are there threshold levels of tau needed to reach a diagnosis of Alzheimer's disease?
Oh, wow, that is an excellent question and a very tough one. More is known about amyloid and those thresholds, and there's just more evidence linking or showing the validity of these amyloid in the spinal fluid or in PET scans with autopsies. There's less known about the tau protein, and I think a lot of that is this technology and the science is still developing, and so the tracers that we use in these PET scans are still experimental, they're still being studied. Our center is one of the centers using them to study their validity, and so I won't be able to answer that truthfully by saying, yes, we know this cutoff and this is what it is. In essence, we're still working at that, and we're still trying to show or prove that what we see in these PET scans is what we would see in an autopsy. The autopsy, the microscope, I mean, that is the definitive diagnosis. So our pathologists are right that is the way we know for sure that a person has Alzheimer's disease. We're hoping that with the advancements in the spinal fluid and in these PET scans, not that we would replace them, but that we could use this information earlier to help people.
Thank you. So the next question, actually, one of Dr. Henderson's slides, really, he didn't had a chance to talk about it, was about oral disease and gum health. And so we had a good question about whether saliva samples could be taken to actually look at gum health and oral pathogenesis, and whether that might, in fact, be another early index for people who are at risk of dementia.
I need my colleagues on either side. Who wants to take that?
I'll briefly say that there have been some new studies linking potentially microbes in our mouth with brain. These studies need to be replicated. Probably the most interesting stuff is actually still in animals, where they find that if they can give a specific microbe, that it can potentially get in the brain and be something that might cause these lesions we see in Alzheimer's, like the plaques and tangles. But it's still a very, very early area, and we need more human studies.
Thank you.
Actually, just one thing to add. When we think about oral health, inflammation, and inflammation being a primary driver of what we think could be contributing to Alzheimer's disease, untreated dental issues are chronic levels of low inflammation. And so that's something that different than the actual microbe itself, but that chronic inflammation that we may be putting off because no one likes to go to the dentist, but in general, that is an important thing to address. And the other thing I'll say is that I watched a 2-minute video from Sanjay Gupta that said one of the 3 things that we can do to live longer is floss, and that's because of gingivitis and oral health.
Okay, so get your colonoscopy and floss. Actually, oral health has been linked to stroke risk. There's some really compelling data on oral health and gum inflammation and risk for stroke and TIAs. Okay, so here's another question about a risk factor, and we had several questions about this. Could one of you please comment on concussions as it relates to Alzheimer's and dementia, and are there any studies at UW being done on athletes who have had head injuries?
There are studies at UW on athletes. There's actually a large study that's being done out of Milwaukee, and UW-Madison is one of the sites, and that's in college athletes, and they're looking at impact of concussion. Obviously not in the context of Alzheimer's, because these are young people. But with regard to dementia, the picture is still mixed. So we know that traumatic brain injury is a risk factor for dementia, but we actually don't know that it's specifically linked to Alzheimer's disease. There's a very large study, the Alzheimer's Disease Neuroimaging Initiative, and in that very large multi-site study, they didn't find that a history of traumatic brain injury was specifically linked with that pathology that defines Alzheimer's disease. But then you can also look at the literature. There's some other studies that suggest that it is risk factor for dementia, unknown if it's plaques and tangles specifically.
So more research needed, right? Mm-hmm. So does hearing loss contribute to dementia?
Yes. Hearing loss is one of those risk factors where, same along with vision, when we think about needing those senses in order to encode information and then recall that information. And actually, there was a study that was just recently published within the past month showing that not treating your hearing or your hearing loss with hearing aids led to an increased risk for not only dementia, but depression and other mood disorders. And so that's a really critical thing that I like to talk about in my clinic, because this is one of the things that we can treat. I mean, we want people to be able to hear. And frankly, if you don't want to hear what's going on, then you just take the hearing aid out. But you have the hearing aid, so I think that's an important thing.
So these are all risk factors that we can deal with. Colonoscopies are good.
Flossing.
Hearing aids. And I don't mean to make so light of it. I think that we tend to forget that there are manageable risk factors that actually significantly reduce our risk of Alzheimer's disease and other dementias. And one of those is diet. So we had a whole group of questions about diet, and I'm just going to kind of, for the sake of time, try to condense them. A, any news on the MIND diet? B, what are your thoughts on a keto diet? And C, are there things that we can be doing earlier in life relative to diet and exercise that would have an impact? So.
All right, so MIND diet, some of you may have joined us for a prior lecture from Dr. Martha Claire Morris at Rush University, and she told us about her work on diet. And in looking at dietary patterns, she found that certain foods were associated with better cognitive function over time. And if you go on the internet and look up MIND diet, Rush University will tell you specifically what those food groups are, but it's high fruits and vegetables, berries, low diets that are low in red meat, cheese, sadly. Don't eat too much cheese. But you can go online and look at those foods. And she's now testing that in a large clinical trial across the United States to see if you start this specific diet, will that be better for your cognitive function? And that's what we need these clinical trials to see. Can you change your diet and have better cognitive outcomes and reduce dementia?
Any thoughts on the keto diet?
So ketone diet, that's a good question. As you'll notice, diet trends swing back and forth. And actually, keto diet is being tested for Alzheimer's dementia. Dr. Henderson mentioned that when your brain uses glucose, but as you develop Alzheimer's disease dementia, your brain actually doesn't use glucose as well. So some people have looked at alternative sources of energy, potentially ketones. However, we're still waiting on results of clinical trials. And so you might be familiar with whatever 3 or 4 years ago everybody said, take coconut oil because that's going to be good for your brain. And that we haven't heard anything about that panning out in clinical trials. But we'll wait and hear if there's some other news.
And believe me, if it comes out to be positive, Dr. Chin will do a podcast on it. I want to put in a plug for his podcast because all of these are excellent questions, and we don't have time to get to all of them, but I can guarantee you that for 90% of the questions that people have turned in, he already has a podcast available online. So I'm going to do 2 more and then do our closing because to be mindful of time. Dr. Gleason, can you tell us about different types of memory impairment? A kind of immediate forgetting, when you go into a room to get something and you can't remember what you were doing, versus other types of memory impairment that are more pathological.
Oh, yeah, sure. This is fascinating. The more I learn about the brain, sometimes the less I understand, I think. It's fascinating. There are many different types of memory, and we've learned about memory because people have volunteered for memory studies, like you all in the clinical core. That's the only way we understand memory, is because a human volunteered for this. So there are the memory, that short-term memory, or that very short-term memory, is something that you might remember over the course of a conversation or immediately after a conversation. Anything longer than 10 minutes, that's long-term memory. So think about that. So it has to become long-term memory, and it has to be consolidated, which means that the hippocampus is involved. So you guys have heard about the hippocampus. It sits kind of right behind our ears here. Think about, this is a link back to that hearing aid question. If you are reducing the amount of input through those auditory processing pathways, it may itself cause some damage to the hippocampus and to those temporal lobes. So that's why that pathway for hearing and language is so important for memory. I mentioned consolidating memory. That's really important. That's how things get stored in long-term memory. And every time you reminisce, you take that memory back out and you re-encode it. So you're recycling that memory and you're re-encoding it, making it stronger. So how many of you have known someone with dementia or with Alzheimer's disease or another type of dementia? I'm sure many in the audience. Could they tell you what happened when they were in the early stages especially? Could they tell you what happened when they were in high school or when they were younger? Oh, yeah. And they might say to you, my memory's fine. I can tell you about where I grew up. But could they tell you about what they had for breakfast? Exactly. Okay. And that is the differentiation there is between a long-term stored memory that is simply re-accessed versus a new memory that has to get consolidated and stored. And that's why we see that difference between that very short-term memory and the long-term memory. So watch out. I can go on forever about memory, so I will stop myself. And yeah, I know. Thank you.
So you wonder, those of you who are participants in our study, you wonder why we do such extensive memory testing, really trying to find those boundaries between what's normal and what's not. And we challenge people. So everyone who comes in for testing will be tested until they fail. That's just the way it's done. So I know, we've heard from our participants that they'd rather do a lumbar puncture than the memory testing. And so we know that it's hard and it doesn't feel good to not be able to answer the questions. But unfortunately, that's intentional.
You're welcome.
Thank you very much. I'll get into that in a second. So one last question. This is one that we haven't ever had before. And that's, can anyone comment on sound wave therapy for Alzheimer's disease? Sound wave therapy. I don't know that we have any.
Yeah, so there's
Ultrasound.
there's a new technology that uses ultrasound. The way that I've heard about it is to actually open up the blood-brain barrier. And if you open up this barrier, you can actually use it to deliver drugs more effectively. So I know that there's developments in that kind of technology. There's also been some work on photostimulation, which could potentially induce brain waves that could clear amyloid. So there is certainly new technologies that are being tested in the context of brain disease.
So for those of you whose questions I didn't answer, we actually take these back and we look for lectures next year, and also we give them to Dr. Chin, who has been really successful in finding people nationally. I don't mean to be so shameless, but his podcasts are recognized by our 32 sister centers as being exceptional. And we have investigators from across the network who have been willing to give him an hour or so to speak. So for those of you whose questions we weren't able to answer, I'd be willing to bet that we probably have some information online, and I'll share these questions to make sure that we cover those topics that we didn't have time to do today. The last question was, do we have enough people to do lumbar punctures? And seriously, somebody actually did ask that question. And so as the person who leads the Outreach Recruitment and Engagement Corps, I'm the person behind all of the asks, along with my colleagues, all of the investigators in the center. And as Dr. Asthana said when he was talking in the beginning, science is hard, research takes a long time, and we can't do it without you. We absolutely cannot do it without you. We could have the smartest people in the world sitting around on a panel talking to each other, and we do love to talk to each other. We really do. But without participants who partner with us to answer these questions, the work goes nowhere, absolutely nowhere. And so I want to thank each and every one of you who has participated in a study, whether it's ours or someone else's. You are driving the science forward. You are, to our good friends from Oneida who drive across the state to do lumbar punctures, I want to send a special shout out to them because our center has been distinguished by your participation. We cannot do this work without you. With these new definitions of the ATN definition that Dr. Chin so eloquently described, because I looked at those tables and it's like, what? We really need people to work with us by participating in our studies. So I cannot thank you enough for coming to hear us talk, to learning more about our work and the research, and for staying with us as you come in year after year after year. When I started this kind of research in 1980, I'm probably the oldest person in the room who's been doing Alzheimer's disease research. I was a new PhD in 1980. We didn't have a diagnostic, we had no diagnostic criteria. Nobody actually talked about Alzheimer's disease. They talked about senile dementia of the Alzheimer's type. And we thought that there would be a cure by 1990. I was at one of the first 10 Alzheimer's centers. It was funded in 1986. We're still searching, but we have a new drug that's going to the FDA that people thought initially was unsuccessful, and then a group of other statisticians put all the data together to figure out that there actually was an effect. So this work is really important, but it goes nowhere without you. So thank you again for coming. I want to thank my colleagues on the panel. I want to thank Dr. Henderson for coming to work with us. His work is incredibly important, and it informs the work that we do here, and risk and benefits for everybody. So thank you so much.